So I would like to start by welcoming everyone to the Cancer Genomics Cloud CGC webinar series. If you haven't used the CGC platform, you can learn how to use it on our website. You can also use it to teach classes and collaborate securely with your collaborators and peers. And you can also follow our innovative data releases as well as apps and workflows. Today's webinar series will be by Trudy Zou. She's on a call. She's going to be telling us about genomic alterations in extraskeletal myxoid chondrosarcoma. Hopefully I did not ruin how to say that properly. Trudy is a medical student at the University of Pittsburgh. She works at the Musculoskeletal Oncology Laboratory with Dr. Rebecca Waters and Dr. Kurt Weiss. And she's going to tell us about an extremely rare patient sample that she has access to in her lab. And I'm just going to pass it on to her to tell us about her research. She started working on the CGC around November last year, and she's done incredible progress, and she's been really great to work with. So floor is yours, Trudy.
Great, thank you, Zelia. I'm going to go ahead and share my screen. Okay, can you guys see the right screen?
You can.
Okay, great. All right, so hi everyone. My name is Trudy. I'm a first-year medical student at the University of Pittsburgh. And today I'm going to be talking a bit about sarcoma. In undergrad, I did 2 years of sarcoma research, and when I got to Pitt, I wanted to continue doing sarcoma research. And as Zelia said, I've only been working on this project for a few months, but I'm excited to share today what I've been able to do so far, exploring extraskeletal myxoid chondrosarcoma. Okay, so first, I'm just going to give an overview of what sarcoma is. Sarcoma are a group of cancers that start in the bone and soft tissues, and they're very rare, and they're also very heterogeneous, and there are over 50 different subtypes. They make up less than 1% of cancers in adults. However, they are much more common in children, and they make up to 15% of pediatric cancers. Sarcomas can be of bone or of soft tissue, and the most common sarcomas are osteosarcoma, which is sarcoma of the bone, chondrosarcoma, which occurs in cartilage cells, and Ewing sarcoma. While osteosarcoma is responsive to chemotherapy, patients with osteosarcoma can still develop resistance, and treatment research is lacking, and many other types of sarcoma, including chondrosarcoma, are resistant to all forms of adjuvant therapy. So this includes radiation therapy and chemotherapy. And for these kinds of sarcoma, the only treatment is wide surgical excision. Sarcoma also have high rates of metastasis to the lungs in particular, and this leads to poor patient outcomes. And because they're so rare, however, they still have high rates of metastasis to the lungs, more research needs to be done to understand sarcomas at a molecular level in order to identify and treat these tumors sooner. There are many challenges to sarcoma research. However, as I said, sarcoma is incredibly rare, so it's hard to collect samples to study, and each subtype is so heterogeneous that sample size is very limited. However, it's still important that we pay attention to these patients with sarcoma, and we need to understand the molecular mechanisms that lead to sarcoma and identify biomarkers that we can use for screening, differential diagnosis, prediction of prognosis and tumor progression to improve outcomes for these patients. And in today's talk, I'm going to focus on extraskeletal myxoid chondrosarcoma in particular, or EMC. EMC is a very rare sarcoma subtype that can actually be in either the bone or soft tissue. When EMC was first discovered, it was categorized as a chondrosarcoma, or a sarcoma of cartilage cells. However, it was actually discovered that EMC doesn't express any of the biomarkers for cartilage. So the World Health Organization actually officially classifies it as a neoplasm of uncertain differentiation, and we're not sure of the tissue of origin for EMC. EMC is characterized by a chromosomal translocation involving the nuclear receptor subfamily 4 group A gene, or NR4A3 gene, on chromosome 9q31.1. And this chromosomal translocation involves an in-frame break with the entire coding region of the NR4A3 gene being translocated to a gene fusion partner. And the gene fusion partner is most often the Ewing sarcoma RNA binding protein 1, or the EWSR1 gene on chromosome 22q12.2, and this occurs in 70% of the cases. This gene translocation results in the EWSR1 gene contributing an N-terminal transactivation domain to the NR4A3 gene, and this leads to constitutive NR4A3 gene expression. The NR4A3 gene can also have other fusion partners. So in 20% of cases, its fusion partner is the TAF15 gene on chromosome 17, and it can also fuse with FUS, TCF12, TFG, and others. And in all of these cases, it leads to constitutive expression of NR4A3. NR4A3 is actually an orphan receptor, and this means that the endogenous ligand of NR4A3 has not been identified. However, the NR4A3 gene controls many different things. It has transcriptional regulatory functions for cell proliferation, apoptosis, DNA repair, metabolism, and differentiation. And it is thought that misregulation in these genes leads to cancer. However, the exact mechanism is still unknown. And actually, in Ewing sarcoma is also caused by a gene fusion with EWSR1. So we know that this gene fusion of the NR4A3 leading to constitutive expression of NR4A3 is key in causing EMC. However, we don't know the exact function or the exact genes that are affected by this gene fusion. So our lab was lucky enough to collect a very rare patient sample of EMC, and we were able to collect the primary tumor, the lung metastasis, the pelvic metastasis, as well as 2 normal samples. We have blood and normal lung, and we were able to whole genome sequence these samples and look for specific biomarkers in this very rare form of cancer. So our patient presented in early 2017. He was a male in the 5th decade of his life, presenting with a mass in the proximal extremity, and this is a pretty typical presentation of EMC. Typically, patients will present with a mass in the proximal extremity, and patient prognoses are usually worse if at presentation they already have metastases to the lungs. And it also has an indolent course. So even if the patient doesn't yet have metastasis to the lungs, or the primary tumor is removed, it may take a while for the tumor to metastasize or for recurrence to occur. So in early 2017, the patient presented with EMC and he was diagnosed and the gene fusion was identified. The primary tumor was removed from his right thigh and he received radiation therapy. However, in 2018, pulmonary nodules were found, and these pulmonary nodules were also removed and the disease seemed stabilized. Later in 2019, there was evidence of slight progression of pulmonary disease as well as recurrence in the right thigh. And also a new nodule was found in the lower abdomen in the iliac lymph node in the pelvic region. And this lymph node was biopsied and it was also found to be EMC. So this nodule was removed and the patient has seemed to be stable since then. And so this is really rare to have a patient, first of all, with sarcoma, and it's very rare that we are able to collect the fresh samples and sequence them immediately, as well as have 3 different sets of tissues from the same patient. In EMC, this is especially interesting because of the indolent nature of the metastasis. We wanted to study the differences between the primary tumor and the lung metastasis and the pelvic metastasis. We expect the mutational burden to increase with each tumor, and we are interested to see which specific mutations may lead to a more malignant phenotype. These images are H&E stains of the tumor. The first image on the left shows the primary tumor, and you can see that there are kind of dark purple spots, and they are also surrounded by more blurry, glassy looking purple, light purple areas. And this kind of looks like what you would expect cartilage to look like on histology, which is why EMC at first was thought to be chondrosarcoma. However, as I said, we discovered that it doesn't actually express any of the cartilage producing cells, and it is a tumor of unknown differentiation. And the image on the middle is the lung metastasis on H&E stain. In the middle image on the left, the area with more white space, that would be the alveoli. And from the right, you can see the glassy and blurry looking tissue invading into the lung tissue, into the alveolar tissue. And finally, on the far right image, that is the iliac lymph node. The top area shows a lot of the B cells in the lymph node, and the tumor is coming up and invading from the bottom. This image it shows the gene fusion product, the NR4A3WSR1 gene fusion product, and this is how we diagnosed EMC. So we can attach fluorescent probes to either end of the EWSR1 gene. The orange probe will give an orange light and attaches to the 5' end of the EWSR1 gene. And the green probe will attach to the 3' end of the EWSR1 gene. And we can image the individual cells biopsied from the tumor sample. You would expect that the orange and green colors are close together because they would be on the same gene. However, when you have this gene fusion product and the EWSR1 gene is split and the chromosome translocation occurs, the green and orange colors will be separated and farther away from each other because they will be separated onto different chromosomes, one still on chromosome 22 and one on chromosome 9. And you can see here that you can see here where the chromosomal translocations occur, where the green and orange colors are a bit more separated. So our aim was to compare genomic structural variants of tumor samples, including the primary tumor, the lung metastasis, and the pelvic metastasis, to our blood, which serves as our control sample, as well as looking at mutations accumulated with progressive metastasis. So our comparisons to look at genomic structural variations of tumor samples compared to blood, our set of comparisons were primary tumor versus normal blood, the lung met versus the blood, the pelvic met versus the blood. And finally, we also had a sample of normal lung, and we can compare that to the blood just as a negative control. And secondly, we also wanted to compare progressive metastases. And so our 2 comparisons that we want to make here are the lung met versus the primary tumor and the pelvic metastasis versus the lung metastasis. And these abbreviations will be used throughout the rest of the slides, so I've explained them here. So DNA was extracted from the primary chondrosarcoma, the lung metastasis, the pelvic metastasis, the normal lung, and peripheral blood mononuclear cells using CIAMP Fast DNA Tissue Kit. samples were submitted to the UPMC Genome Center for further preparation, sequencing, and analysis. And libraries were prepared via the Illumina Nextera DNA Flex library kit, and samples were sequenced on the Illumina NovaSeq 6000, and 30x coverage was used to obtain greater than 1.6 billion 150 base pair paired end reads for each sample. For analysis, the Dragon genome pipeline was used to map reads to the human genome assembly. We wanted to look at three main kinds of structural variations from our whole genome sequencing. And these three categories include single nucleotide polymorphisms, copy number variants, and structural variants. And these essentially vary in their size. So first, MuteCT2 software was used to identify single nucleotide polymorphisms. And as this indicates, single nucleotide polymorphisms are usually variants of just one base pair or one nucleotide. We also looked at copy number variants, and we used CNV-KIT to look at copy number variants. And this just looks at if any genes are amplified in number or decreased in number. And finally, we wanted to look at structural variants, and we used the 7 Bridges Cancer Genomic Cloud platform, and specifically certain callers, including Delly and SVABA, to look at structural variants. Structural variants are usually greater than 30 base pairs. However, this is a bit of an arbitrary definition, and this includes shared patterns of structural variation. And structural variants, or SVs, are actually understudied compared to single nucleotide polymorphisms because they're harder to identify. They can be blurred by sequencing and mapping errors, and structural variant patterns can cover a large portion of a read or be larger to read length than the read length, which complicates mapping. And patterns induced by different structural variants can also be very similar or be overlapped or nested. So these are a bit harder to identify, but in the past few years, there have been a lot of great programs that have come out that make it much easier to identify structural variant patterns. And many of these platforms are hosted. There is an app on the 7 Bridges platform from which we can use these different platforms and look at structural variants in our genomic sequencing data. So first, I'm going to talk about the single nucleotide polymorphisms or variants, SNVs, found in our EMC samples. So this is an oncoplot of the different genes that were impacted the most by SNVs in our EMC samples. Starting off on the x-axis has each of the comparisons that we were looking at. And from left to right, that's primary tumor versus blood, lung met versus blood, pelvic metastasis versus blood, and the normal lung versus the blood. And on the y-axis, we have genes that have a lot of SNVs found within the genes, and they are ordered from top to bottom, with genes being most impacted in all of these samples at the top and genes impacted less at the bottom. And the different colors represent the type of SNV that was identified in the gene. For example, you can see a lot of green, and green is a missense mutation. There's also a lot of the lighter red color, which is the nonsense mutation. And there's a lot of black, which are multi-hits. You will also notice that the third column, which is the pelvic metastasis versus the blood sample, we actually found during our analysis that this comparison has an abnormally high amount of mutations, much higher than we would expect. And this could be due to the fact that the patient received radiation therapy before we found the pelvic met. It could also be due to a sample preparation error or an analysis error. So we are rerunning the analysis for that sample now, and we hope to be able to add it into our analysis in the future. Taking a closer look at the different SNVs found in each of our comparisons, this shows a zoomed in version of the oncoplot shown on the previous slide. So in looking at this data, we want to focus on the primary tumor versus the blood comparison and the lung metastasis versus the blood comparison. And this is because we want to look at the genes that are most impacted by mutations, because these mutations may be in genes that are key in misregulation or key in causing EMC. So we found a few genes that are interesting and that were highly impacted by SNVs, especially in our primary tumor and our lung metastasis. And these were mostly impacted by missense or nonsense mutations. So first of all, we found FRYL, or Fry-like transcriptional coactivator, and OR5F1, and these genes actually both play roles in development of neuronal sensory fields. And this is interesting because, as I said previously, EMC is actually a neoplasm of uncertain differentiation. However, research in the past few years has indicated that EMC might actually be of neuronal tissue origin, which is very different from what we originally thought. However, this matches a lot of the current research in the field, and would be interesting to further explore to see if EMC is in fact of neuronal sensory tissue origin. IL-7R, or interleukin-7 alpha receptor, is also interesting. Mutations in ILR7 contribute to leukemia, and it is actually targetable by the JAK-STAT pathway inhibition because it interacts downstream by the JAK-STAT pathway. And this gene is an interesting example of a possible biomarker for EMC. So if our patient responds to certain treatments, such as JAK-STAT inhibitors, this could be applied to other patients that also have EMC that also might have mutations in this specific IL-7R gene. And finally, UBD, or ubiquitin D, also known as FAT10, is also interesting. Some studies show that UBD stimulates osteosarcoma, which is a different kind of sarcoma of the bone. And in one study, silencing UBD inhibited metastasis of osteosarcoma. Next, we just quickly looked at copy number variants in EMC, and this CNVs, or copy number variants, indicate if certain genes have been amplified in copy number or decreased in copy number. This plot shows different comparisons, or 4 comparisons from top to bottom: the lung met, the normal lung, the pelvic met, and the primary tumor, all compared to blood. And the x-axis shows each chromosome, and the red color indicates that the genes in that chromosome, there are more genes that have copy number increases. And the blue color indicates that there are more genes in that chromosome that have copy number decreases. So here we were just curious to see which chromosomes were heavily impacted by copy number variations. And in the future, we can look and see which specific genes within these chromosomes were contributing to the copy number increase or decreases. So again, looking at the primary tumor and the lung met comparisons, chromosome 1 and chromosome 8 have copy number increases in the lung metastasis and the primary tumor. And chromosome 6 and chromosome 10 have many copy number decreases in the lung metastasis and the primary tumor. So we'd like to focus in the future on these chromosomes and see if any of these copy number increases or decreases in particular contribute to a malignant phenotype. And chromosome 8 in particular has more copy number increases in the lung met than the primary tumor. So it would be interesting to see if these genes specifically contribute to an even more malignant phenotype of the metastasis of the tumor. Finally, we looked at structural variants, and in EMC, it's especially important to look at structural variants. So structural variants, as I said, are larger scale mutations or patterns, and generally greater than 50 base pairs. And because EMC is defined by a chromosomal translocation, it's especially important to look at these large scale variants in EMC. And translocations are not identified by the other two methods. So here I am again illustrating our known transposon in EMC. And this is the NR4A3 EWSR1 transposon or gene fusion product. In looking at structural variants, we use different callers that make comparisons between the tumor sample and the control sample or the blood sample. And it looks for structural variants. And when running our callers, we want to select the callers in particular that can identify this translocation that we already know and expect to find in our samples. And so we will call these patterns V1 and V2. So V1 would be the first half of chromosome 22 translocated onto chromosome 9, the second half of chromosome 9, and V2 would be the first half of chromosome 9 translocated to the second half of chromosome 22. So this is our NR4A3 ESWR1 gene translocation products. So we know that callers that can identify these translocations are probably more reliable in our samples. So we ran these callers, and because I had just hopped onto this project a few months ago, I haven't yet had time to look too deeply into the different patterns identified, but I will just give a brief overview of what we have so far. And we also specifically looked for patterns that were found in the lung metastasis versus blood comparison, and saw if any other comparisons shared these patterns. And so we were able to find 4 patterns that were found in the lung metastasis versus blood comparison that were also found in some other comparisons. And we're interested in looking at SVs found in the lung metastasis compared to the blood in particular, since the lung metastasis is currently our most malignant phenotype. So the 2 callers that we ended up using that were able to identify our known translocation were Delly and SVABA. And Delly found 2 patterns that were present in the lung met versus blood comparison, and SVABA also found 2 patterns. So in Delly, the 2 patterns found, we'll call them P1 and P2. P1 was our known gene translocation product, as well as a deletion on chromosome 10 and a deletion on chromosome 19. And P2 was our known gene translocation product, but the endpoint was slightly different. In SVABA, we have 2 patterns, and we'll call them P3 and P4. P3 was our known gene translocation product, as well as a deletion in chromosome 10. And P3 identified by SVABA might be the same pattern as P1 identified by Delly. P4 was a more complex pattern, and this is an example of something that might be more complex that is not identifiable by SNV or CNV analysis. So P4 included mutations in chromosome 10, 2, 4, 8, and chromosome Y. So these are our results of looking at structural variants in EMC First, our sequencing results were run through callers such as Delly and SVABA, and these made comparisons between our different tumor and normal control samples to identify structural variants that were present in the tumor samples and not the control samples. Then we put the results of these callers through an app on Seven Bridges platform called Conseca. And Conseca can identify if there is a pattern that is present in one set of comparisons that's also present in a different set of comparisons. So if we first look at the first plot on the left, this is the results from the Delly caller. On the top, we have the intersection size of the structural variants found. And in this case, translocations are actually just identified by the nucleotide that they start at, which is why the size is smaller than we would expect. On the y-axis on the bottom, we have our different comparisons. So for the Delly plot, from top to bottom, these comparisons would be lung met versus primary tumor versus normal lung, lung met versus normal lung, primary tumor versus blood, and lung met versus blood. And these comparisons are ordered by the comparisons that are the least similar. So here, the lung met and the primary tumor are more similar, and the lung met versus the blood have greater differences. And like I said, we first wanted to ensure that these callers could find our known gene translocation, and we denoted our known gene transfusion products as V1 and V2. And in the Delly plot, you can see that the orange circles indicate that our known gene translocation is present in those samples, or that V1 and V2 patterns are present. And so we can see that kind of in the columns at the right of the plot. The vertical lines between each dot indicate that this pattern is shared among the different comparisons. So this means that the gene transfusion product is found in the primary tumor versus lung comparison, the lung met versus lung, the primary tumor versus blood, and the lung met versus blood. So it is present in all of the tumor samples and none of the control samples. And it is also not present in the top row, which is the lung met versus the primary tumor. And it does not show up there because these samples both have the translocation. So the difference would not be detected. And this is the same with the SVABA caller. We can see V1 and V2 are identified in all of the comparisons, comparing a tumor sample to a normal sample, whether that be blood or lung normal. And the gene transfusion product is not identified in the sample comparing both tumor samples, or comparing the lung met and the primary tumor, since they both contain the translocation, so this difference would not be detected. And this highlights that the gene transfusion product is present. And next, we were looking at pattern 2, which is our gene translocation product, but slightly shifted. And we see that it is present in the lung met versus blood comparison. So this means it's present in the lung met sample and not the blood. And we see that it's also present in the lung met versus primary tumor comparison. So this means that it's likely present in the lung met, but not the primary tumor. So we want to look for patterns like this because they are present in the lung met and not the primary tumor or the normal tissue. So they could possibly contribute to a more malignant phenotype in EMC. And these types would be more important to target because in the end, metastasis to the lungs leads to worse patient prognoses, but patients with stable sarcomas generally do well. Pattern 4, or P4, is the more complex pattern identified by SVABA. and it involves structural variants in chromosomes 2, 4, 10, and Y. And again, we see here it is present in the lung met versus the normal sample, and it is also present in the lung met versus the primary tumor sample. So this means it's likely present in the lung met and not the primary tumor or the normal sample. So this also would be interesting to further explore. So from here, we would like to take a deeper dive into the structural variants identified and see which genes are specifically impacted by our structural variants. And so I'll just give a quick overview of the data we have, and we're hoping to look further into the genes, these specific genes impacted in the next few months. So here we have the structural variants in the primary tumor, found in the primary tumor compared to the blood sample. The x-axis indicates the chromosome number, and they are sorted by which chromosome has more structural variants identified, and the size of the structural variants is on the y-axis. We normalize the structural variants by chromosome length, since we expect longer chromosomes to have a greater mutational burden and therefore more structural variants. And so we were looking at which chromosomes have an abnormally larger amount of mutational burden compared to their length. So this graph shows the structural variants normalized by chromosome length. The top row denotes the chromosome number. The y-axis indicates what kind of structural variant is present, and we looked at break endpoints, deletions, inversions, and duplications. And the yellow color indicates a greater number of structural variants, and the black color indicates less structural variants normalized by chromosome length. So we can see here that chromosome 19 has a particularly large number of structural variants, as well as chromosome 16, chromosome 11, and chromosome 10. It would be interesting to look and see which genes are specifically impacted by these structural variants. And it also looks like duplications and deletions in particular are highly present in our primary tumor sample. Next, we have the lung metastasis sample. And again, the chromosome number is on the x-axis and the number of structural variants is on the y-axis, and they're ordered by chromosomes with the larger number of structural variants. And again, we have these normalized by chromosome length. And in the lung metastasis, there were mostly inversions and deletions. And in the lung metastasis, we see that, again, chromosome 19, chromosome 10, and chromosome 8 have many structural variants present. So wrapping up, our aims were to look at structural variants or mutations on many different levels. We first used MutCT2 software to identify single nucleotide polymorphisms, or SNPs. And from this, we found a few different genes that had SNPs. These included FRYL and OR5F1, which are involved in neuronal sensory development and could inform us of the true tissue origin of EMC. It also included IL-7R, which could be a possible targeted biomarker, and UBD, which has been found in other studies to possibly stimulate osteosarcoma metastasis. Next, we looked at copy number variants, and we found that there were many copy number increases in chromosome 1 and chromosome 8 in our tumor samples, and there were many copy number decreases in chromosome 6 and chromosome 10 in our tumor samples compared to our normal blood samples. And finally, we looked at structural variants. We were able to identify our known genes translocation, which was our known gene translocation, and using the callers Delly and SVABA. And we also started looking at other interesting patterns, and specifically we were looking at patterns that were present in the lung metastasis, but not the other samples, to see if these patterns might be particularly malignant. We also saw that chromosomes 8, 10, and 19 have increased numbers of structural variants, especially in the lung met, and also comparing to CNV analysis, chromosome 10 has high numbers of both CNVs and SVs. So in the future, it is important to further explore the structural variations and patterns identified through Delly and SVABA and look specifically at the genes impacted by these structural variants. We would also like to add the pelvic metastasis comparison to study mutational progression of metastasis. Metastasis is a progression that starts from micrometastasis, and generally the cancer cells become less and less like the original cell as they metastasize. And studying metastatic progression is especially important in sarcoma, because the metastatic sarcomas are the ones that lead to worse patient prognoses. And so we would like to identify targetable biomarkers or molecular profiles for EMC. In doing so, we can better identify possibly which patients, depending on their molecular profile, are more likely to met out later and target these patients with therapy. In conclusion, there are a lot of barriers to sarcoma research, including an extremely small sample size and just a very heterogeneous mixture of sarcomas. The pathogenesis of EMC and other sarcomas is very poorly understood, and this makes it difficult to identify biomarkers and develop therapies. So it is important to expand our very small database of EMC knowledge. No predictive factors currently exist to predict metastatic disease and inform the use of systemic treatment. And we are wondering if it is possible to identify therapeutic markers that will predict if EMC will eventually metastasize, so we know which patients we should start on therapy. And molecular profiling of EMC can help us develop targeted, personalized treatment for individuals. So that is all I have for my presentation. I'd like to thank the members of my lab in the musculoskeletal oncology lab, and especially Dr. Kurt Weiss and Dr. Rebecca Waters, my PIs. Thank you to Dr. Ivy John for providing pathology images, and Anish Chaka Numachandran with UPMC for help on the bioinformatics analyses. Also, thank you to the UPMC Cancer Center Biospecimen Tissue Core, and this work was in part funded by the Shadyside Hospital Foundation. And finally, I had a lot of help at Seven Bridges and a lot of support, especially hopping on the past few months and getting to work on these technologies and being able to access my apps so that I could run my callers. So thank you to Poro and Dave and Anna at Seven Bridges.
Thank you so much, Trudy. That was a wonderful talk.
No problem.
You did great progress in the past couple of months working on this. We do have one question at the chat. I believe you answered the second question our participant had. So the first question is, are there metastasis tissues extracted after the treatment? And if so, will the treatment affect the mutations in the metastasis tissues?
Yes, that is a great question. And that is actually one that we were wondering too. So as I mentioned, our pelvic samples, we ran the analyses and we found that there was an especially high mutational burden on the pelvic sample. And because we had treated our patient in 2017 or 2018 with radiation therapy, this probably contributed to the high mutational burden in the pelvic sample. So we're currently rerunning that. And as for the primary tumor and the lung metastasis, it's definitely possible. But we can focus on the mutations or the structural variants that are in common between the lung met and the primary tumor. because the primary tumor, we extracted the sample before radiation therapy.
Okay. Thank you. Yeah, that makes sense. So during your talk, you mentioned in the conclusions that you wanted to look at gene expression. So what other kind of sequencing technologies will you planning to implement? Since these samples are so rare, will you be able to do RNA-seq or not? Do you not have enough sample for that?
Yeah, I think gene expression is something we'd like to look at eventually. Currently, we're just focusing on the genomic structural variants. Maybe in the future, we might see if we are able to run RNA sequencing, if we can get more sample. I actually don't think we have enough of the pelvic sample, but that's definitely something that's important to look at, especially because EMC actually, it has these larger gene translocations, but it has a higher mutational burden despite most of these having the main translocation being a very large one, as opposed to other cancers that have a lot more small mutations that contribute to their phenotype.
Yeah, that makes sense. I kind of figured that would be the answer. It's such a rare sample. How do you do your results compare to the other sequencing reports in EMC, if there are any?
Yeah, so actually, let me see if I can go back. Our copy number variation, the copy number variations in these chromosomes, and like copy number increases in chromosome 1 and 8, and copy number decreases in chromosome 6 and 10, there are similar copy number chromosomes found in the literature in the past. There's like not many studies done overall, but these actually have been found in past literature. So it's good to see that our data matches. And also in terms of like the single nucleotide polymorphisms, we also found that many of the genes that were implicated in our data contribute to sensory neuronal field development. And this has also been found in a lot of other studies. So yeah, we have a few overlaps with other studies in the past.
That's wonderful. I guess the next big question that I have, if there are not any more questions from the participants, is I was wondering how did the CGC help with your project, and how difficult or how easy was it to use?
Sure. It was, so I do have like a little bit of background in bioinformatics, and I've used Python and R in the past. And because of that, I do know how difficult it can be to use these platforms that have been recently developed all by yourself. And right now there are so many tools that can be used, and there are so many callers. We use like Delly and SVABA, but there are so many more like NovaBreak and Manta, and Seven Bridges actually already has a lot of these apps on the platform. And it's really easy. You can just download the app and click on it and put in your input files, and it spits out the output files. And you can like adjust the different settings by just like drop down menus. So it really is like so much more easy in terms of the apps that are really accessible. And then I think also what made it really easy was the people helping me, Poro in particular, is just really, really responsive. And if I like have a question, or if I want to use an app that's not on the Seven Bridges platform yet, him and Dave Roberson have been very helpful in helping me create that app on Seven Bridges so that I could use it.
That's great to hear. Glad you had a great experience. So if there aren't any more questions, we can end this webinar a little bit early. I just wanted to let everyone on the call know that we do have office hours, and you are always welcome to contact us. As Trudy just said, we are very responsive. So if you need anything, you have more information about the Cancer Genomics Cloud, you can always email me or go to our website at cancergenomicscloud.org. Or attend our office hours that are on Tuesday at 10 a.m. and Thursday at 2 p.m. Eastern. So thank you so much. And if you have the chance, if you want to watch it again, we have this recorded. Thank you so much again, Trudy.
No problem. Thank you.
Thank you so much.
Great day. Bye.